So before we begin, I'd like to acknowledge the people of the Kulin Nations on whose land our institute is located and pay our respects to elders past and present. So today for the Monash Advanced Microscopy seminar series, we have Lukas Kapitein, who is from Utrecht University. I'm sorry if I butchered that, in the Netherlands. So Lukas Kapitein studied experimental physics and graduated in biophysics. During this time, he fell in love with motor proteins and the cytoskeleton. Ever since, his research is aimed at understanding the mechanisms underlying intracellular transport and spatial organization within cells. To understand the interplay between cytoskeletal organization and intracellular transport, his group has developed multiple innovative super-resolution approaches and pioneered the use of optogenetics to control intracellular transport. So Lukas will be talking to us today about super-resolution microscopy of cellular organization in health and disease. So thank you, Lukas, if you'd like to begin.
Thank you, Jessica, for the introduction, and thank you all for the invitation and for coming to listen. So I'll be talking indeed about super-resolution microscopy of cellular organization in health and disease. So my lab has a long-standing interest in understanding the mechanisms by which cells establish their shape and intracellular organization. And we mostly focus on neurons because arguably these are the most beautiful and most complex cells in our body. And so what we try to understand is how it establishes its shape and complex organization, and also how it manages to have a single axon that can be up to a meter long, as well as multiple dendrites that have a completely different morphology and also a different function. So the dendrites receive signals from other cells, and then based on these inputs, a signal can be transmitted down to the axon. Now, this complex organization and these differences between dendrites and axon is established because proteins that are synthesized in a cell body can be differentially transported either to the dendrites or to the axons. And this is made possible by the cytoskeleton, which you see here, the microtubule cytoskeleton of the neuron. And this functions as a road system for motor proteins to deliver transport either to different parts of the cell. And so this is a long-standing interest of us. How do we build specialized cells? And also more recently, we also started working not only on neurons, but on other types of differentiated cells, such as airway cells or other epithelial cells. Now, one other very interesting aspect about neurons is that most neurons you're born with, they will also die with. So how is it possible that we can have cells in our body that live for so long? For example, the gut epithelial cells, they are renewed every week. And so there's a completely different challenge for neurons to stay alive for so long. And of course, many neurodegenerative diseases emerge because of the emergence of toxic protein aggregates. So the idea is that one reason why we can keep cells healthy for a long time is because our quality control system and aggregate clearance system is very active in scouting aggregates and removing them, and that this deteriorates over time. And this is something we became recently very interested in, and I'll also tell you a little bit about this. So these are the biological questions that we ask, but we also ask methodological questions. Because in order to understand how the cytoskeleton, the microtubule network, guides motor proteins to specific destinations, we really need to map them at a very precise resolution. And so we're developing a lot of we're spending a lot of our time in developing new tools for super-resolution microscopy, but also for live cell imaging to really map cellular organization and dynamics across scales. And also, if you image a lot, it's very beautiful, but what we in the end are after is mechanistic insights in what causes which behavior and how are things organized in the cell. And it's not always easy to dissect cause and consequence in complex systems. And therefore, we found actually that the optogenetics can be very useful to have much more well-controlled experimentation in cellular systems. I'll also show an example of that. So in this seminar, I will cover 3 topics: microtubule organization and neuronal transport. So for our core interest, I'll spend about 20 minutes on that. Then a recent effort to go with our super-resolution microscopy not just from cells cultured on glass to really tissue systems. And then finally, I will spend a bit of time on this recent work on aggregates and how they impact nuclear integrity. But first, transport. So as I explained to you, the microtubule network in neurons is used by motor proteins to drive directional transport. The microtubules are polarized structures, so they grow head to tail from subunits like Lego, and this gives the whole polymer an asymmetric nature, an intrinsic structural polarity with a minus end and a plus end. And this is recognized by motor proteins, so many motor proteins from the kinesin family move towards the plus end. This is like a large family of different motor proteins that mostly move to the plus end. And then there are the dynein motors that move to the minus end of microtubules. And so, because there are many different families and that cargos can recruit different types of motors, and not just microtubule-based motors, but also actin-based motors, this leads to very big differences in the dynamics of different cargos in the cell. So here we see the dynamics within dendrites of secretory vesicles, late endosomes, lysosomes, or recycling endosomes. And you can really see that they really have very different dynamic. And we want to understand like what is the origin of these differences in dynamics and what's also the function of this difference in dynamics. And this is partly the origin of this difference in dynamics, partly due to the different motor proteins that are recruited. But one very confusing or interesting aspect of the microtubule network in dendrites is that they have mixed polarity. So in the axon, all the microtubules have a uniform orientation. All the plus ends are pointing outward. And so when you recruit kinesin, you know that you will have anterograde transport away from the cell body. And when you recruit dynein, you will have retrograde transport to the cell body. But it's not so easy in dendrites because there's a mixed microtubule organization. So even if you activate one type of motor, you cannot really predict which way they will go. And so this is something that puzzled us for a long time. How can you get regulated transport in a system where the underlying microtubule network appears to be random? Now, in order to understand how different motor proteins navigate the microtubule cytoskeleton, we introduced a number of very well-controlled systems where we can look at the activity of specific motors. And this uses inducible heterodimerization system. So we started initially with the FKBP-FRB system. These are two engineered protein domains that will bind together upon the addition of a small molecule called Rapalog. So we fuse one of these domains to a motor protein, of which we deleted the cargo binding domain. So these are constitutive active motors that will just walk, but they don't carry cargo. When we add Rapalog, we can recruit these motors to the cargo of interest. In this case, we use peroxisomes. So here you see the distribution of peroxisomes. And during this video, we add Rapalog to recruit kinesin. It will bind to the peroxisomes and it will direct the peroxisomes along towards the periphery of the cell. In this case, it's a trivial outcome because we know that in these cells, microtubules have a radial organization with a minus end in the center of the cell and the plus ends at the periphery of the cell. A bit more recently, we also used optogenetics, where we use light-sensitive heterodimerization. We use a protein domain that's found often in plants that can change configuration upon illumination with blue light, and that triggers then the dimerization of these domains. And there we can use pulses of light to recruit the motor to the peroxisome and then induce transport. And when we stop illuminating, this is reversible, so the transport will stop. One beauty of the use of using light is that you can apply light very locally, so you can illuminate one part of the cell and only induce transport in that part of the cell. And so we use these type of assays for a number of applications. First of all, probing the dynamics of specific motors, so we can really test all the kinesin family members one by one and see how they behave and what their preferential destination is. We can also make combinations of motors and see how they influence each other's behavior. And we can establish custom organelle distributions to really study what's the function of different organelles at different locations. And so this is something, a number of projects in the lab that are along these slides. But for now, I'll just show the result of recruiting specific motors two peroxisomes in neurons as a tool to see what is the preference of these motors. And we found broadly two different behaviors that I will illustrate with this video. So this is the behavior of kinesin 3. This is a plus-interacted motor protein. And if we recruited the peroxisomes in neurons, we induce transport. And the transport goes both into the axon as well as into the dendrite. And you cannot see it in this video, but I will tell you this. In the dendrites, these cargoes actually accumulate in the tip of the dendrite. So despite the microtubule network having a random organization, this motor somehow is able to navigate from one side to the dendrite all the way to the tip of the dendrite, which is remarkable given the mixed microtubule organization. Now, a different behavior we saw is the behavior from kinesin 1. And here you see that this is also a plus-interacted motor. It also drives anterior transport into the axon, but it is completely not entering into the dendrites. So here we have two quite similar motors, both are directed towards the plus end of microtubules, have completely different behaviors within the dendrites. And this has been a long-standing goal of the lab to understand how this is possible. And the underlying assumption that we had, or the hypothesis, was this encoded in the organization of the microtubule cytoskeleton. So I'll just give you the model that we came up with and then show you the evidence that supports this model. So a key ingredient of these models is that microtubules are not all the same in neurons. It's well known, and I'll come back to it later on, that microtubules can be either dynamic, so they grow and shrink, or they can be more stabilized. And when they are more stabilized, they are long-lived and they accumulate multiple post-translational modifications. And so within neurons, we find different subsets of microtubules. And it's also known that different motor proteins preferentially interact with different subsets. And so the hypothesis that we had, and it turns out to be true, is that within the dendrites, we have mixed microtubules, but that one subset of microtubules is predominantly oriented minus and out, and the other subsets predominantly oriented plus and out. And because they recruit different motors, this can explain why some motors actually can enter the dendrite and even accumulate in the tips despite the mixed orientations, because they see a microtubule network that is mostly plus and out. And then other motor proteins like kinesin 1 that like to bind to stable microtubules, they see a microtubule network that's largely minus and out, and therefore they will not be able to enter the dendrite and only enter into the axons. This is now our current model for transport in dendrite, and I will try to convince you that this is correct based on the data that we have acquired. This is what I was telling about microtubules. Microtubules are dynamic polymers, they grow and shrink, but in neurons there is a large population that somehow is stabilized, and because they are long-lived, they accumulate these different post-translational modifications that supposedly changes the function of the microtubule, perhaps by recruiting, by changing the structure or recruiting microtubule-associated proteins that can recruit or inhibit binding of different motor proteins. So here you see this illustrated. In red, you see acetylated microtubules. These are long-lived microtubules that are stable and have acquired this modification called acetylation. And then tyrosinated microtubules. These are microtubules that have freshly polymerized, and here their C-terminal tyrosine has not yet been cleaved off, because detyrosination is the modification that happens on polymerized microtubules. So here we can really, by just staining for these two different groups, we can really distinguish the two different subsets in neurons, and we can already see that they have a really different organization and they barely overlap. And also within the neurites, here we see indeed that there is a core of stable microtubules surrounded by more dynamic microtubules. So the next question is, to support our hypothesis, what are the orientations of these different microtubules? And this required to have a tool that allows us to tell what the absolute orientation of microtubule is. And so here we see a microtubule end, but we cannot really tell whether it's a minus end or a plus end, because in neurons there is no centrosome that centrally organizes microtubules, as you also can appreciate in this image. And so therefore we established a new tool, and this tool is called motor paint. So what do we do here? is we can fix cells in such a way that the microtubule network is really nicely preserved. And then we can add purified multiproteins. And these purified multiproteins, they can still bind to the microtubules of this fixed cell that we extracted. So we removed the membrane and we washed away most of the cytosol, but the cytoskeletal is still there. And these added multiproteins, they can still, in presence of ATP and magnesium, they can still bind to microtubules and then walk along microtubules. And now we can use the whole toolbox from single molecule localization microscopy. We can locate all these micromotors with nanometer precision and then just acquire many, many frames. And from these videos, we can reconstruct the microtubule network that's used by these motors. And because the motors bind and walk towards the plus end, we can even color code the microtubules to indicate their absolute orientation. So in this case, we look at microtubules grown from a central microtubule organizing center, and we color code them based on their orientations. And when they are red, the microtubules are oriented towards this corner of the image. When they are blue, towards this corner, green, this corner, yellow, this corner. In this case, it's trivial that all the minus ends are in the middle, and then all the plus ends are pointing outward. Here we see another cell, the periphery of a cell, also mostly plus ends at the periphery of the cell. But for example, if we zoom in on this box here, we see here two microtubule ends, and one is clearly a plus end, whereas the other is actually a minus end, which you wouldn't have been able to tell by just doing super resolution. But using Moses paint, we now can probe the absolute orientation of microtubules in a whole network. So when we do this in dendrites, we can confirm indeed that they have mixed orientation. So this is the super resolution image based on all the localizations. And here we have color coded the microtubules based on orientation, and we find that approximately half of the microtubules is minus end out oriented and the others is minus end in oriented. And we see also that there are tyrosinate microtubules and acetylated microtubules, that they have sort of non-overlapping organization. So now what we can do is we can add nocortisol, which is a drug that depolymerizes microtubules. But it will only depolymerize the non-stabilized microtubules. So we lose the tyrosinated microtubules, but the acetylated microtubules remain. And then we can repeat multi-paint and again reconstruct the microtubule network and quantify the orientations. And in then this situation, we find that the majority of microtubules that remain after the cortisol treatment are oriented with the minus end pointing outward, confirming that the stabilized microtubules are the microtubules that are minus end out, whereas then the dynamic ones are oriented mostly plus end out. Because we know that kinesin 1 prefers stable microtubules, kinesin 3 dynamic microtubules, this explains why one motor can enter the dendrites, drive directional transport, whereas another motor cannot enter the dendrite. Now, so this is quite an interesting insight, but still it's a bit limited because we do this all in 2 dimensions, and we became more and more interested to understand microtubule specific organization in 3 dimensions. And here localization microscopy is not necessarily the best approach, and we started to turn to expansion microscopy, which was at that point just introduced by the lab of Ed Boyden. And this is a very exciting approach where instead of trying to get improved resolution by improving your microscope, improving the optics, or making fluorophore switch from dark states to illuminated states, instead you find a way to physically magnify the cell so that you get improved resolution in your image. It's like making a drawing on a balloon and then blowing up the balloon. You get a much more crispier image of your drawing. Of course, you cannot just do this. You cannot just blow up cells and think that everything will be fine. So you need to do it in a very controlled way. And they, Chen, Tilburg, and Boyden introduced this approach, which depends on the use of swellable hydrogels. So these are gels that when you add water, will absorb the water and thereby swell up. This can also be found in baby divers, or you have sometimes this dinosaur eggs that you put in water, and then the dinosaur inside will swell up and crack the egg and born. And so the idea, you can actually use an acrylamide gel that you also use for normal protein gels, but you add sodium acrylate, and the sodium acrylate makes this a hydrogel. So the trick is to make sure that your proteins, or especially your antibodies and fluorophores of interest, are covalently linked to the gel. And then you add water to swell the gel. But before you do that, you first denature all the proteins of interest. So you really make, you fix the cells, you stain them, and all the antibodies and proteins you cross-link to the gel. And then before you swell the gel, you denature the proteins to make sure that the protein structures inside the cell do not resist swelling. And so actually the final sample that you have is mostly water and a small peptide fragment with fluorophores that are cross-linked to the gel. And so it sounds almost too good to be true that this is really possible, but we tried it out and we were quite amazed that it works pretty robustly. And here you see the microtubule network in neurons, now not just in 2 dimensions, but in 3 dimensions. And you see that all the microtubules are very nicely linear polymers, so they didn't seem to be cracked and broken. So it seems to work pretty well. And in this case, we reach an expansion factor of 4. That's the classical expansion microscopy approach. So when we do this in dendrites, we can stain for tyrosinate microtubules, acylated microtubules, and also for total tubulin, because it will allow us to quantify the relative contribution of these 2 subsets to the total microtubule array. And I hope you appreciate from these videos that actually the organization in 3 dimensions is even more remarkable than 2 dimensions, and that we keep seeing a stable core, a core of stable microtubules surrounded by a shell of dynamic microtubules, which mostly are polymerizing close to the plasma membrane. And here you see it also in a cross-section. So this is all the microtubules in a cross-section, tyrosinated microtubules, the dynamic ones, and the stable ones. And so you clearly see a non-overlapping organization. We can make radial density profiles for different microtubules, get an average of multiple dendrites. So what's the relative distribution across the radius of this dendrite? And what we can also try to do is then say, can we actually get back the density of total tubulin by a weighted sum of acylated and tyrosinated distributions? And this is sort of possible to a relative extent by weighting them in a factor of 2 to 1, suggesting that we have twice as much stable microtubules as dynamic microtubules. It's sort of an estimate. We could take this one step further. So the resolution of a light microscope is of course best in the XY direction, whereas we are interested in the cross-section of a dendrite. So what we can do with these gels, we can, after swelling, we can cut them, flip them, and we can image them then with the highest resolution in the plane of interest, which is then the cross-section. And we can really start to see individual microtubules within these very dense dendrites. And so we did an analysis and we tried to really identify individual microtubules and measure the level of tyrosination and acetylation. And we found for the microtubules that we find in the tyrosinated channel that they are to some extent acetylated, but not a lot. And that the acetylated microtubules that we find in the acetylated channel, they are actually barely tyrosinated. But there is some crosstalk, which makes mostly chemical crosstalk, so that the antibodies actually still recognize the other subset of microtubules because there might be a certain degree of modification. But now that we know this for individual microtubules, we can actually use the same technology as what you use with spectral demixing and tease out the absolute number of microtubules, because we now know the intensity of individual microtubules, which are tyrosinated or acetylated. We can analyze the total intensity divided by the intensity of individual microtubules, get an estimate for the number of acetylated microtubules and the number of tyrosinated microtubules, and also the relative distribution. And what's quite remarkable, it seems that we can account for all microtubules that we identify in the total tubulin stain by a sum of the acetylated microtubules and the tyrosinated microtubules. So it seems that there is really 2 major classes of microtubules, stable, dynamic, and we can also count them. And again, we find that there are 2 times as much stable microtubules as dynamic microtubules. And we can also make an estimate of the density. We find 55 microtubules per square micrometer, which means that there is a microtubule every area of 130 square nanometers, which is relatively consistent with the estimates based on electron microscopy. So we are quite confident that we do our counting correctly. So this is recent work that we published where we really try to be very quantitative about microtubule organization in dendrites. So to summarize this part, I've told you about selective transport in neurons to the axons in dendrites, our advances in mapping the microtubule network, so going from stat to expansion to this flipped expansion approach. And now what we really want to understand is how do you build such a complex microtubule network where there is stable microtubules in the center, dynamic microtubules surrounding it, but also stable microtubules mostly oriented minus and out, and dynamic microtubules mostly oriented plus and out. In addition, we want to understand how multiproteins can be so selective for different subsets. What guides that kinesin 1 only binds to the stable ones and kinesin 3 preferentially binds to dynamic ones. And how does this all impact microtubule organization and organelle dynamics? And one major goal for the coming years is actually to map microtubule organization, not just in these cultured neurons, but really in tissue neurons. I'll show our first steps towards this. So to understand this question of how microtubules are actually built, how this microtubule network emerges, we have spent quite some time to develop a marker that can selectively label this subset in living cells. So far, all our identification of stable versus dynamic was based on fixation and then staining. But of course, that this gives you a very static picture, and it would be very, very nice if we can visualize it in living cells. And so we just, the paper came out a week ago in Journal of Cell Biology, where we introduced actually a mutated motor protein that really nicely labels a subset of microtubules, and we do all the controls in the paper and show that this is a bona fide marker of the stable microtubules. So hopefully in the coming years, this will allow us to learn much more about how these microtubules emerge, and especially how they get their stereotypical organization within the dendrites. So one project we already did with this marker is just one illustration of you can use this marker also beyond studying neurons. Here's an example of cell division, where you beautifully see that just after anaphase and on the onset of cytokinesis, you get this massive accumulation of stable microtubules in the midzone, which was already known based on fixed experiments, which we can now really follow the time course very precisely, and it's very stereotypical. We also see that when the cells enter interphase again, that we get this very stereotypic interface area of stable microtubules. So I hope this tool will be widely used by the community, and you can also find it on Agile. So one other project we did with this is to combine actually fluorescence microscopy and cryo-electron microscopy, because we were interested in the idea that perhaps stable microtubules have a different structure, a different lattice structure than dynamic microtubules. So it's well known that microtubule stabilizers, such as Taxol, they change the lattice spacing of the microtubules from 8.2 to 8.4 nanometers. And we were thinking that perhaps there are also biological mechanisms of stabilization that can change the microtubule lattice. And for this, we needed, of course, to use cryo-electron microscopy. But then how can we identify the stable and dynamic microtubules? Because you cannot do stainings in these type of samples. And so here, stable marks is very useful because the cell is already expressing the marker in a fluorescent way. You can do cryofixation, cryofluorescent microscopy, then do fit milling, but you can try to correlate the fluorescence image that marks the stable microtubules with the electron microscopy image that shows you all the microtubules, and you can analyze the microtubule lattice spacing. And this was done in collaboration with the cryo-electron microscopy group from Stuart House in Utrecht University. And indeed, they found that stable microtubules that are decorated with this marker for stable microtubules, have an expanded lattice compared to dynamic microtubules. So I think this is a very interesting structural insight into the differences between this different subset of microtubules, and also illustrates nicely the combination of fluorescence microscopy and electron microscopy. But this is so far all done on cultured cells, cultured on glass, which of course is quite an artificial system. And if we really want to understand neuronal organization of tissue and also perhaps how this is altered in models for neurodegeneration, we really need to move more towards tissue. And so if you want to move from cells to tissue, you have a number of problems. So you have much more background. The tissue is much more scattering, and so the wavefront is aberrated. So your quality of imaging is less precise. The C resolution becomes very important because you have a thick specimen. And there is a problem of antibody penetration. So it's much harder to stain your samples. So initially, we were trying to solve this problem still using single molecule localization microscopy. And we introduced the use of adaptive optics to study this much better. So we've published a manuscript where we show an optimal way of correcting the wavefront based on the blinking of the molecules. So in localization microscopy, you have a lot of blinking molecules, and based on these blinking molecules, you have to estimate what is the aberrations in our samples. It is not trivial, but it can be done, and then you can really see a big improvement when you actually optimize one of the deformable mirrors in the system, and it gives you a much better pointed function, which you then can use for localization microscopy. And for example, we can really reveal this very stereotypic actin spectrin repeat in the axon initial segment at the depth of 50 micrometers in brain tissue. But it's not a very trivial method to use, and also in localization microscopy, multicolor imaging is still a challenge. So here we also search to use actually expansion microscopy much more. And we started collaborating with one of the inventors, Prof. Tilburg, who just was playing with finding new recipes that would give an increased expansion factor. And so this is also published recently, but together we managed to find a recipe that was really optimal for subcellular imaging and provided an expansion factor of 10. And so I will spare you the details, but the key step is actually to reduce the cross-linker concentration. So when you have your less cross-linkers, you have the less chance to have this cross-links between polymer strands that can impede expansion. And so when you decrease the cross-linker concentration in the gel, you get higher expansion factors. But of course, at some point, you no longer have a gel. So there is a trade-off. And so we found that 10-fold expansion is sort of the best, the place where you should stop trying to get for more. And this works beautifully. I will just now show a few slides with some examples. Here in blue, we see a total membrane stain that also led to plasma membrane, the microtubule network in 3D, and then here the endoplasmatic reticulum stain labeled with 661. And here's another example where we use a plasma membrane stain that nicely labels, in this case, the microvilli of CARCO2 cells that form a nice brush border. And these are a hollow, you can clearly see in this video that these are hollow, and the dimension is 100 nanometers. So our resolution is really, maybe below 50 nanometers, because we can clearly see that these microvilli are hollow. And I think compared to, for example, electron microscopy or scanning electron microscopy, the big advantage is that you can really get the volumetric information from just imaging one step of a cell. So you can do optical sectioning and get the whole volumetric ultrastructure of this cell. Here's another example of airway epithelial cells. These are cultured from nasal samples from human subjects. You can grow them, differentiate them in the lab, and you get a very beautiful epithelial cell layer with this cilia. And in this case, we use the total protein stain. This is actually was introduced by other labs, where you just add fluorophores that can covalently link to lysines or cysteines, and therefore you can get also ultrastructural contrast. And you can beautifully see the basal bodies and these interdigitations between cells. You can also combine the total protein stain and the membrane stain, and then again, video playing. You can really beautifully render these airway cells in 3 dimensions. You see the ultrastructure, and you can also render it as if it was scanning electron microscopy or transmission microscopy, but it's all based on the same data set. So this was a beautiful starting point to then start to go into tissue. Here's one illustration. Let me see if this video is playing. Okay, that seemed to be a problem. I hope you just saw the ultrastructure, and perhaps there's another slide. I think my PowerPoint is overloaded. Okay. Here's the video. So this is a video where we stain the synapses. So Homer and bassoon. One is the presynaptic, one is postsynaptic. And you see that in this piece of tissue, there is thousands of synapses, but you can beautifully resolve the presynapse and the postsynapse and the synaptic clefts. We can also combine these stainings, get ultrastructure and selective labeling in one sample. So now, of course, the big question of expansion microscopy is you get beautiful pictures, but how do you really know for sure that what you see hasn't been distorted by local anisotropic expansion? How can you be sure about the exact expansion factor, and how can you be sure that there are no deformations? So this was something that was bothering us, and we wanted to get a robust way to address this. And at some point we found a solution, and it's illustrated in this picture. So what we can do is we can actually create cover slips that have a patterned proteins. So here in yellow, you see proteins that have been micro patterned on glass. And then cells can still grow on this, and they are not hindered by these proteins because we coat it with another protein layer. And so then when we prepare our samples for expansion microscopy, these proteins will just also be embedded into the gel, be digested, and still we can image their fluorescence. So we can really get a reference system, a reference grid of known size that will tell us the exact expansion factor, but also whether or not there were deformations. So here we see a bigger picture of the same grid used after expansion, and you see clearly that there has been quite some deformations on long length scales. So these lines, they were originally straight, but after expansion, you see that they have been deformed. And for example, the boxes here are much smaller than the boxes there. So the expansion factor here is actually much higher than the expansion factor there. But because we know the original pattern that we used, we can actually computationally correct this. So we now know for every place on the cover slip what has been the deformation in the gels, and we can correct for this. And that's also what we've done in this picture, this corrected picture based on the gel. Of course, you can think, well, maybe you should just stick to the middle of the cover slip, and there the deformation is not so pronounced. But also there you sometimes find regions that are beautifully preserved, and other regions where you have sometimes rupture, but also more local deformations. And so based on this grid that you will always have on your image, because it's been transferred from the cover glass into the gel, you can now do quality control. And you can either decide not to image a certain place, or you can image it and then use the grid to correct the image. And this also works on tissue, because then you make a tissue slice and you just put it on the cover slip during gel, and then do the gelation. And then it will also, the grid will be embedded in the biological sample. And in this video, you see that the grid also has been deformed, but we can use the ground truth here to correct the whole tissue image based on the grid that we have imaged. And then we can zoom in, and we can really see the blood vessels, and then all the neuron within this piece of tissue. So GelMap, we call this method GelMap, and it facilitates both calibration, and we measuring the expansion factor, but also the correction of deformations in expansion microscopy. Here you see again this illustrated. We now can really do imaging across scales, go from whole tissue into subcellular structures like here the nucleus. And we know we can do quality controls. We know the exact expansion factor. and we know that we don't have massive deformations. One other use for this method is to do correlative live and expansion microscopy, because this is quite a challenge if you do live cell imaging and then you want to do super resolution of the same cell. This is in principle possible, but if you do a distillation step and a swelling step, then the cells detach from the cover slip. So it's not so trivial to find back the cell that you have been imaging. But GelMap also provides a coordinate system, so we can do A1, B, C, B2, so we can really find the cells that we image, because the coordinate system is also transferred to the gel. This is illustrated here. We image a mitotic cell, and it's lying in this close to the L box, and that allows us to cut out this piece of gel and then image that piece of gel on the microscope and get the ultrastructure of the cell that we have also imaged live, as you can illustrate in this video. So that is the second part. So now and we are really currently trying to push this for brain tissue and to really stain cytoskeletal structures, microtubules, dynamic stable microtubules in brain tissue, to see if these structures and this microtubule organization that we identified in culture neurons, whether it's preserved and whether perhaps different neuronal subtypes have different types of microtubule organization. Now, in the final few minutes of my talk, I will just give you a small glimpse of what we are doing in terms of studying aggregates. And I need to introduce this a little bit. How did we get into studying aggregates? This is because at some point we wanted to develop for our inducible transport assays, we didn't want to use peroxisomes anymore, but we wanted to have a much more orthogonal cargo. So we started to play with making protein clusters inside the cell that would then serve as cargos for motors. This was a bit naive because, I mean, we could make protein clusters in the cell using homodimerization and heterodimerization, as illustrated here. But it was clear that these were not more inert than the peroxisomes, because the cells actually started to clear them. So they would already be mobile without recruiting motor to them, because they were a target of autophagy. And so actually we put this to good use. So we labeled them with both GFP and RFP, and we could make this into a sensor for autophagy. Because inside lysosomes we have acidic pH and a lot of proteases, and so proteolytic proteins. And so that actually, but the M-cherry is not affected by those. So GFP will be rapidly quenched and also degraded, but M-cherry will actually resist this. And so when we see a conversion from yellow to red, we know that they are transferred to the lysosomes, and we can measure autophagic flux of these aggregates. And then in some experiments, we targeted them to the nucleus, and we found that having aggregates in the nucleus was actually impacting the integrity of the nucleus. And of course, this is a very highly artificial system, so we decided to study this for real pathological aggregates, and we focused on the Huntington protein, because it's known from mouse tissue and other pathology experiments that they often form also in the nucleus. So it's what you can do with that. So Huntington protein has poly-Q repeats, and depending on the length of the repeat, the protein will aggregate or not, and you will get Huntington's disease or not. So when you have only 23 repeats, you can express the protein, you can target it to the nucleus, it will be diffuse. But when you have 74 repeats, then it will aggregate either in the cytosol or when you add the NLS, in this case, in the nucleus. But it's in pathological samples, in histology samples, also without the NLS, the full length Huntington protein will actually be present in the nucleus. Now, what we were then doing was imaging the integrity of the nucleus by using a stable cell line expressing RFP NLS. So this is a protein now targeted to the nucleus. And what you find is that sometimes the nucleus would actually rupture. You see it here as a flash. So I will repeat the video. So you see first this blabbing, and then you see that the RFP leaks out of the nucleus, which indicates that there has been a rupture in the nuclear envelope. And this is, of course, problematic, but the cell, in principle, is able to repair this very quickly using escort proteins, and therefore then the original distribution is restored. But we found that, well, you always have a bit of ruptures in cells, but when you have these aggregates in the nucleus, you have much more ruptures. So almost 1/3 of the cells in our experiments would undergo a rupture. But what's even more dramatic is the following. We often found that these ruptures would occur, as illustrated here, but they would not be repaired. So somehow in the presence of these aggregates, the nucleus was unable to repair itself. And so this bill is very interesting. Of course, it can have huge impact because the cell, in principle, tries to keep the nucleoplasm and the cytoplasm separated from each other. And so here's the quantification. Okay, so here's the quantification, so that in normal cell, wild-type cells, all the ruptures are repaired. But when we have the aggregates in the nucleus, we see that in half of the cells, the nucleus is not repaired, and that in the cells that can repair the nucleus, this repair is actually much slower. So the recovery of the RFPMLS into the nucleus is much slower. So I'm apologize for my PowerPoint. A few more slides. Okay, I might have to exit this and go to the slide directly. So again, we use now to understand why in the presence of aggregates we would have a lot of failure to repair the nuclear envelope. We started again to use expansion microscopy, tenfold expansion microscopy. And I think this illustrates beautifully the power of this method, because you can now really see much more structure in these aggregates within the nucleus and even in 3 dimensions. And so what we could identify is places where the nuclear envelope have been ruptured. So here we see the lemon matrix, and we see that these fibrils are actually sticking through the hole in the matrix. And also here we look at the total membrane stain, and we see an aggregate, and here we see perhaps an attempt to repair this ruptured membrane, but you see that the fibril is in the way, and we see a lot of membrane, but perhaps there is still a leakage. So this brings us to the following model for what's called an Arminius system. That in the presence of nuclear aggregates, we see a lemon destabilization. I didn't show this data, which can lead to blebbing and a rupture. And because these are fibrillar aggregates, that are very often the fibrils will actually start to stick out, and this will impair the normal repair with the escort proteins, and therefore we get membrane accumulation, but not really a healing. And therefore, we have a long-term loss of nuclear integrity. So we now currently are following up these and studying much more the cell biology of this. So this concludes what I wanted to tell. I talked about microbial organization, neuronal transport, super resolution microscopy, moving towards tissue, and the role of aggregates on nuclear integrity. This I would like to conclude. Thank you for your attention. I apologize for some failed transitions in the PowerPoint. And I want to point out that we have several interesting positions available. For example, a microscopy specialist for our more custom systems. So we're looking for somebody with a strong background in establishing custom setups. But it's also interesting to train a lot of users on more conventional setups. And it's an interesting position because it's a permanent position. Also, we have various PhD and mostly postdoc positions on various aspects that are presented. And we're looking for assistant professor in cell and neurobiology, as well as a lecturer in biophysics. So if you're interested in any of this, please write me an email and we can have a call. Thank you very much, and I'm happy to answer questions.
Okay, thank you, Lukas. So we have time for some questions now. So if anyone from the Zoom wants to ask a question, you can just use the raise hand function, and then we can unmute you and you can ask your question, or you can pop it in the chat and we can ask it for you. So I might start with a question myself. So I was interested in the motor paint technique, where you can kind of reconstruct the microtubule network by looking at the tracking of kinesins, I believe.
Mm-hmm.
So I was kind of wondering if that was how that would differ, and if it was maybe a more accurate way than where you can use tracking of end binding proteins like EB3 or EB1. So how would that kind of differ to tracking it that way?
Yeah, so in principle, in living cells, if you want to understand microtubule polarity, microtubule orientations, you can indeed use the tracking of EB proteins, which bind to the growing microtubule placents. And in that way, it has been, for example, shown that in dendrites, you have twice comments growing outward and comments growing inwards. But if you count them, you find twice as much growing outward than growing inward. Whereas based on early electron microscopy, the estimate was that the microtubules had a 50-50 mixed orientation. So there was sort of a discrepancy between these EB experiments and the EM experiments. And that's because the EB experiments only track the end of the microtubule and they don't report on the length. So if you have many short microtubules growing outward and one long one growing inward, then in every cross section you have 50-50, but when you count the ends, you overestimate the ones that are placent out. Also, it's important to realize, and that's also what we has been known from other work, but also we confirm it in our recent paper on the microtubule stable marker for microtubules, is that many stable microtubules in the cell, they don't even have a growing placent. So if you use EB to measure orientations, you are biased towards the subset of stable microtubules, and you might miss a subset. So I think that's why we developed motor paint, because we want to have a method that probe microtubule orientations and microtubule mass in one method, so that we and would not be biased towards one subset or another.
Mm-hmm. Okay.
It's a good question. So in the first order method, if you want to just probe, is my network uniform or mixed? Then probably if you only find comments moving outward, then it's a good indication that it's mostly uniform, but you have to realize that you might miss a subset of stable microtubules that don't grow and might have an opposite orientation.
Yeah, okay. So we have some questions from the audience. So the first one, I'll ask Pete Curry I think Stephen can unmute you.
Can you hear me?
Yes.
Yes.
Oh, great. Fantastic talk. Really thought-provoking. And the last couple of slides, you showed aggregates, nuclear aggregates forming or perforating the nuclear lamina. And I just wondered whether you'd had an opportunity to image that dynamically. I'm interested in the actual mechanism of perforation, or whether it's actually a disassembly of the lamina or incorporation of the lamina proteins in the aggregates. Do you have any idea about how that actually occurs?
Yes,
I've seen this one. Yeah, go ahead, sorry.
Yeah, it's a good question. So, I mean, I showed only a bit of the data. So we actually pre-printed the work. I'm not sure if I put the reference here, but you will be able to find it. But what we find most often in this video and also previous, that the rupture is preceded by a bleb. And I think that blabbing suggests that that we are looking at the destabilization of the lemon matrix.
Yeah,
So
I mean, exactly, yeah, because it's very reminiscent of what you see in Emily Dreyfus nuclei or laminae mutant. So I wondered whether it was a weakening rather than a perforation somehow. That's kind of whether there was more dynamic interplay than just simply
exactly. Exactly. I think it starts with a weakening. That's also in our cartoon. So that we think this initial step is the weakening of the lemon matrix.
Right.
We tried to look for phosphorylation. I think we couldn't really see it, but we see already without ruptures, we see an impact on lemon organization. Right, and then once you have a bleb and a rupture, then that just gives way for the aggregates for one of the fibrils to just pop out.
Yeah, so there's a regulatory event at the lamina that's facilitated by the aggregates, basically.
Exactly, and that I must say, and this, what if it's biochemical or physically that it's sort of a disruption, because also the lemon, I think also had the LAM proteins in the nuclear envelope, they also seem to be connected in some way with the chromatin. So it could also be that somehow just the integrity of the nuclear envelope and its association with the chromatin is disturbed. So we are actually, so if you have any good suggestions of experiments to test, we don't know if it's directly lemon destabilization or if it's just sort of interfering with the linkage between the nuclear envelope and the chromatin and the lemon.
Yeah, I wondered
physically by intercalating.
Yeah, my only suggestion is if you could actually simultaneously image the nuclear lamina like beta-1 or A dynamically and at the same resolution, I think that would be a fascinating way.
Yes. So we have done it, it's in the manuscript, but we haven't done it at super resolution, but with normal lifestyle imaging. And there you also see often like transient accumulations, and you also see scarring. So that sometimes when there has been a rupture, then you see a scar, so you get a massive accumulation of lamins. And sometimes you also see, yeah.
Very cool, Lukas. Thank you. I'll look up the paper.
Okay, thank you. Thank you for your question.
Okay, and the next question will be from Sebastian. And I think Stephen can unmute you.
Hello, can you hear me?
Yes.
Yes.
Hello?
Yes,
Hello?
we can hear you.
Yeah.
Can you hear me
Go
now?
ahead.
Yes.
Oh, okay, perfect. Sorry, my microphone was not working. Thank you very much for the interesting talk. I guess my question is about this relationship you mentioned between the orientation of the microtubules and the post-translational modifications that they have. So which one do you think has the bigger influence on the other one? So is it, the orientation that influences how are they modified afterwards, or the other way around maybe?
Yeah, it's a good question. And so the short answer is we don't know. But I think it's pretty hard to conceive how an enzyme that does modifications can be selective for one orientation over the other. We are now analyzing really how this network emerges, like in 1 day of culture, even 6 hours of culture, 12 hours of culture, 18 hours of cultures. And already from the beginning, we see directly after plating that microtubules, that the neurons have stable microtubules and dynamic microtubules. And so we think that the modification of the microtubules precedes their orientation. And then the question is, okay, how can you make sure that stable microtubules have an orientation that is minus and out? And there we are currently looking into the role of kinesin 1, because kinesin 1 is not just a motor that moves over microtubules, it's also a motor that can organize microtubules. So there's, especially from the Galvin lab, there is a number of papers that show that kinesin that's anchored to organelles or the cortex, it tries to walk over microtubules, it walks to the plus end, but in fact, it pushes the microtubules with the minus end leading. And that could be actually the mechanism why this specific subset that is recognized by kinesin 1 is oriented minus and out, because the motors actually push them in minus and out. So that's currently our thinking.
I see. Well, thank you very much.
Yeah, but great question. I mean, this is something we really want to understand. That's why also we spent this time in developing this marker for stable microtubules, so that we hopefully during development can see like how this organization emerges.
Okay, so we also have a question from the chat. So the question is from Hanadi. Hanadi, you're welcome to jump in and read it out yourself. If you'd like to, just raise your hand, or I can read it out for you. So we have the question. They've said, thanks for this very interesting talk. You mentioned staining for stable microtubules, but are there stains to stain dynamic microtubules?
Yes, so maybe to explain it so extensively. So microtubules, when they are polymerized, they have they are polymerized from tubulin, and free tubulin has a C-terminal tyrosine. And when after polymerization, and when the microtubules are long-lived enough, then there is an enzyme that cleaves off the C-terminal tyrosine. So if you stain for tyrosination, you stain for microtubules that presumably have just been polymerized. So that's why we all the time compare tyrosine and acetylation, because the modification that happens on the microtubule is detyrosination. You can also stain for detyrosinated microtubules, but they largely overlap in this system with acetylated microtubules. So we assume that the tyrosination is a stain for recently polarized microtubules.
Okay. I also have another question on your the stable marker of the stable microtubules. So one of the challenges that we sometimes have looking at microtubules using fluorescent microtubule associated proteins is if we overexpress them, they can actually stabilize microtubules themselves. So does the stable mark, like how does that actually bind to microtubules and how does that work?
This is it's a great point, and if you, I mean, the paper just came out last week, so you'll see that we do many controls, and we show that if you express it too highly, you get bundles of microtubules. Mm-hmm. This is also a major concern of the reviewers, of course, that that if you overexpress it, you have too much. So how do you know what's the right level? So that's why we made a stable cell line expressing this marker. And we were checking for, we were really looking at, okay, control cells in the level of acetylation, and then cells expressing our stable mark and the level of acetylation. And we see that if you overexpress it, you get more stable microtubules, more acetylation. So we actually went all the way towards using FCS, fluorescent correlation spectroscopy, to measure the concentration of stable mark in cells that don't show defects. So we have sort of a concentration range of expression that you should use to not have artifacts. So it's all in the paper, but and we were quite happy that we were able to get a stable cell line that went through mitosis and show no mitotic defects. And actually that the stable microtubule network is disassembled on the onset of mitosis, it's re-emerges. So therefore we are fairly confident that when used properly, it will not cause too much defect. I have to point out that, as you also point out, this is true for every fluorescent marker. Mm-hmm. Also, EB3, which everybody uses to label growing microtubule ends, I mean, it changes the structure of the plus end. So if you overexpress EB3, you actually change the dynamics of microtubules. If you use LifeAct, you also, if you stabilize the microtubule. So every marker that you express in a cell, that you overexpress, will change the organization. So it's a good point, and you should be very careful.
Yeah, I'll have to check out that paper. Yeah. Okay, so we might leave it there. Thank you so much for sharing your work with us, Lukas. It was really extremely interesting. You've created such a powerful toolbox of ways to see the microtubules and to understand their dynamics. And thank you for waking up so early in the morning.
Now it's live, so it's fine.
Yeah,
it was my pleasure. Thank you very much for the invitation. If you have any other questions or are interested in any of these positions, please feel free to email me.
Great. All right. Thank you. We might leave it there then. Thanks, Lukas. Thanks, everyone.
Bye-bye.
Bye-bye.